Why do I have to go to school?
What should I be learning?
What is education?
The true tradition of the origin of the word education comes from Old Latin for ducare, which means to pull out, which is very different from to lecture, which is to pump in knowledge.
The quality of education can never exceed the quality of teaching. There are no schools better than the teachers, because great teachers make people learn better and become great learners.
The teacher has to be a person who inspires people to get motivated so that they can become problem solvers. And that changes the role of a teacher from being an authority figure to being a facilitator. And so I see the role of a teacher as an agent of change.
One of the most exciting things about being a teacher is when you're working with students and you're sharing your passion or your excitement about what we're learning about. And students are kind of sitting there and there's a lot of confusion, and then all of a sudden it all makes sense, and you can see it in their eyes, you can see the excitement.
The next instrument I would like you to know, students, is wind vane. As you can see it here, it's made of metal. It comes in various sizes. But this typical wind vane I'm holding here has 4 cardinal points. This is the north. This is the south. I hope you can see it.
Yes, sir.
I hope you can see it, students.
Yes, sir.
This is the north, this is the what? South. This is the west, and this is the what? East. Then this is called the arrow or the pointer. So you carry this wind vane and place it in the field or in the environment where it cannot be disturbed. Then as the wind blows, the arrow moves. The wind moves it. The wind moves it. So in any direction that this arrow is pointing towards, for instance, students, tell me, which direction is this arrow pointing now?
Southeast.
South what? East. This is southeast. It means that the wind is blowing towards what? Southeast. So any direction that arrow is pointing becomes the direction of the what? Wind. Therefore, wind vane is an instrument used to measure the direction of wind what? Blow. We also have another instrument we use in measuring wind, that is called the anemometer. That one shows the wind speed or the speed of the wind. So the next instrument we shall talk about here now is the test tube rack. Although I have shown you before, please pay attention here.
Yes, sir.
The test tube what? Rack. This is a test tube what? Rack. This is a test tube rack. This is a test tube. This is the rack. So we use this test tube rack to hold the test tubes, to keep them so that they will not break easily. We use them to set the test tubes out so that we can observe. The next one is the tripod stand. This is a tripod stand which we have already demonstrated, and other associated structures that goes with it. So this is a tripod stand. It has 3 legs. That's why it's called tripod. Tri means thread. You can see it here. We normally place it over the Bunsen burner during heating to serve as a platform over which the beaker or the flask will be what? Placed for heating. We normally place wire gauze over it and the porcelain triangle to support it, to support the beaker or the flask to be heated. The next one is the incubator. Yes, you can see the incubator here. We have incubator here. This is a very small sized incubator, but we have bigger one we use in the hospital, but this is a small sized incubator. You can see it, the inside. So this incubator can be operated electrically. It can be switched on and set to a particular temperature for the growth or culturing of some biological specimens. For instance, if you want to culture bacteria, we place it in the incubator for about 24 or 48 hours. And after that, we bring the specimen out and observe the growth of the microorganism. So incubator is very good. As we move ahead, we shall see how to use this incubator in culturing of microorganisms. Okay, the next one we shall look at is the plant pots. You already know that one, plant pots. Yeah. This is the plant pot, yes. We call it plant pot because we normally use it to keep plants growing in the lab. If you look inside, you see fertile soil. It's a normal garden soil. And this is the plant that is growing after it has been germinated in the pot. So with this pot, we shall be able to monitor the growth of the plant on a daily basis. We observe changes that occur in this plant under laboratory conditions. So we want students to know exactly the real nature of the plant pot. It comes in various sizes. This is just one example of a plant pot. As we go ahead, we shall demonstrate how to use this plant pot in biological experiments. Then the next on the board is the insect net. We have different types of insect nets. You can see this one. It is just like a very long spoon with net and a handle. When we use this insect net, we scoop the environment to catch insects, flying insects especially. We use it as an ecological instrument. We use it for catching flying what? Insects. We shall soon demonstrate how to use this insect net in the field. When you enter the field for ecological survey, you will use it to scoop the surface of the vegetation or the grasses, and you scoop insects inside it, and the insects are later transferred to the containers and preserved for biological examination. We shall also see the crucible tongues. This is a crucible tongue. This is what is called a crucible tongue. It's also like scissors. And this is called crucible. This is a crucible. Please look up. This is what is called a crucible. It comes in various sizes, and we use crucible for heating purposes, for heating solutions, for heating reagents. And this is the tongue. Sometimes after heating the crucible, after heating it, it will be very hot, that you cannot touch it with your what? Hand. Am I correct? Yes. If you heat this thing and it's very hot, can you touch it with your ordinary hand? No. So for you to lift it up or for you to bring it out from the burner or from the steam bath, you use crucible what? Tongue. And when you use it to hold it and transfer it to another position where you can observe what? Changes. So this is what is called the crucible what? Tongues. We also use it to pick other hot objects during heating. Then the next is the supporting ring. This is the supporting ring. You can see it here. This supporting ring is used to support the hot crucible. After the specimen or the sample or the solution has been heated and the crucible becomes very hot, you can use the crucible tongue to pick the crucible and transfer it to the crucible, to this ring. We call it supporting ring. So use this, the crucible tongue, transfer the crucible to the supporting ring, and observe changes in the solution you have heated. So that is the importance of the crucible what? The ring support, or the supporting ring. Then the next one is the test tube holder. We have talked about it. Then the triple, please look up here, look up and watch closely. This is called the triple beam balance. Triple beam balance. Why do we call it triple beam balance? It has 3 parts here for the measurement of masses in grams. You can see the calibration. This is from 0 to 10 grams. This is from 0 to 500 grams. This is from 0 to 100 grams. Depending on where the mass of the specimen or the substance lies. When you place, for instance, if you want to weigh this section of orange, if you want to know the mass, you place it here. When you place it, you begin to adjust, do some adjustments here until it comes to a point when it will balance, when the beam balances with the weight of the what? Orange. Then you cannot take your readings. We shall demonstrate how to use this as the class goes on. The next on the board is the watch glass. This is the watch glass. This is called the watch glasses. They are also glasswares, but highly breakable. We use it to wash and observe specimens, sorts, changes that occur on a particular specimen or sort. If you want to see it clearly, especially those that have eye problems, that find it difficult to observe changes. So we use watch glass, place the specimen and observe it. For instance, as we shall carry out soon, experiments on how to test a leaf for starch. At the end of the experiment, before we apply iodine solution, we are going to place the leaf on the watch glass and apply iodine. You will immediately observe a color change. So watch glass enables us to watch any change in color of a specimen or a sample under investigation. So it is a vital glassware in the lab. The next is the spatula. This is a spatula. Please pay attention this way. This is a spatula. And this spatula is used to collect samples of salts or chemicals from a bigger container where you cannot put your hand, because you are not expected to dip hand into a container to collect chemical or samples or salts. So we use spatula. Open the bottle. For instance, if this is copper 2 tetraoxosulfate 6, this is copper 2 sulfate, and I want to collect samples of the copper 2 sulfate. You are not expected to dip your hand into this bottle, so you use spatula and collect this salt. You collect this salt and transfer it to the solution where you want to dissolve it. So that is what the spatula is all about. I hope that point is very clear.
Yes, sir.
We can now move ahead to the next, which is the dropper. You have droppers here. Droppers comes in various sizes and shapes. Look up. This is called what?
Dropper.
What did I call it?
Dropper.
Now this dropper is used to collect samples of solutions for dropping during chemical reactions, or for dropping, for mixing with another solution. For instance, if you want to collect few drops of a copper 2 sulfate solution, I can use dropper. Watch me. You put this dropper, press it, press the head, the rubber end of the dropper, press it. As you insert it, then leave your hand. It is going to draw some liquid. Can you see it?
Yes, sir.
Then I have collected some liquid drop. Then this liquid drop can then be transferred to the appropriate reagent or solution you want to mix it up with. So that is why it is called what? Dropper. You collect the solution and drop I hope that point is very clear.
Yes, sir.
Then we have water or steam baths. The water we always, this is the base. This is the base or the platform where you can place beaker or conical flask. So this retort stand is always for the suspension of conical flasks or burettes, so that liquid can pass down to the flask or the conical flask placed on the base. So this is the clamp. You can raise it up to any height you want it, depending on the length of the burette or the funnel you are holding it with. So this is what we use in holding funnels or burette. For instance, this is a testo funnel which you can easily place here and use the clamp to hold it. So when you clamp it in this position, it is going to stand. Then you suspend it. If there is any reaction that you want to observe, or if there is any liquid in this funnel or burette, you can easily pass it down while you keep this glassware on the base of the clamp. So we shall demonstrate how to use it. As this practical class goes on, we shall demonstrate how to use it. Then the next is the testo funnel, which I have already shown you. This is a testo funnel. It has a very long narrow tube with again a wide mouth. The mouth is open. This side is open, this side is open. But we can cover the mouth if we want to pour solution into it for obvious reasons. So as we go ahead in our practical demonstration of osmosis, then I will tell you why the mouth should be closed or covered. But this is just a funnel. We use it doing experiment in osmosis. We also use testo funnel doing experiment in transpiration. Then we shall talk about the specimen bottles. We have many of them. Specimen bottle is a bottle we use to preserve specimens. If you look at this specimen bottle, it contains some specimens like cockroach and rats, and it is always labeled. The specimen inside it is labeled. The chemical is formalin or formaldehyde, which we use to preserve the specimen and keep them in their lifelike condition. At times, you get specimen for examination and you don't want the specimen to decay or decompose. So you use the specimen bottle to preserve the specimen with the preservative. You can see it here. We have other specimen bottles that contains other specimens. So specimen bottle is mainly for preservation of specimen. Then we have refrigerator. We have refrigerator here for preservation of specimens as well as fruits, if especially when they are not needed immediately. We have stop clock. The stop clock is used for timing. It's a second. It counts in seconds, so we use it for timing during biological experiments. So as we go ahead, we shall demonstrate how to use the stop clock. We have the masculine tape. This tape is used for labeling of specimens. It has a gummy surface. So when we label any specimen, we now attach it to it with this masking tape. So it is very important in biology lab. For labeling is very necessary, because when you don't label a particular specimen or a particular chemical or reagent, it becomes so dangerous to deal with such situations. So that is why we always label. Labeling is very important. Tweezers, I have talked about them. These are tweezers. They are found inside the dissecting kit, and we use them for lifting, picking tiny specimens and the cover slips during microscopy. Thank you.